Welcome back to the Imprint Hub, where we talk about maternal and pediatric pharmacology. Today, we are talking about lactation, and specifically lactation at the cellular level. We are joined by Dr. Britt Goods, who's going to explain to us how studying cells from breast milk can help us better understand milk production and drug exposure during lactation. And with that, I'll turn it over to Dr. Goods.
Okay, so today I'm going to be focusing on how single cell methods can be used to understand human lactocytes. So, what I hope you'll learn from this talk today, that cells in breast milk can be used to study the lactating mammary gland, and these cells are highly dynamic. I'll also talk about how we can use single cell methods to understand these cells and their basic biology. And then I'll also talk a little bit about some newer lab in my work where we're currently using these cells to perform small molecule perturbation studies. And so I'll walk through each of these sections as a part of this talk today. So to start, I think for folks generally interested in this, the mammary gland itself is quite dynamic, as well as breast milk that's produced by the mammary gland. And so over here, I'm showing an image from a really beautiful review paper that talks about all of the different things that can actually influence the mammary gland and hence milk production and the components in milk. And these are can be quite broad. So really big things like maternal health status and behavior, for example, all the way down to sort of cell-based, on the cell-based and sort of molecule scale. So for example, like genomic variation as well as hormonal regulation. And then over here, I'm showing a really nice image from a review article that talks about the developing mammary gland. And so just to give a high-level overview of what that looks like. So the mammary gland is comprised of these ducts, which have these little lobules at the end that during lactation will actually fill with milk. But during puberty, these are sort of already templated. These structures exist in the breast. And then in response to hormonal changes during pregnancy, these structures will dramatically expand and change in the breast. And then, of course, fill with milk during lactation. And then through the process of involution, sort of return sort of to baseline, sort of to baseline. And so pictured another way, I really want to zoom in on the cells. So I think this image does a really good job of showing sort of the overall changes that can happen in the breast. But I think this image does a nice job of showing the cell types that are actually important and involved in this process. And so again, here I'm just showing an image, a cartoon that shows what a duct might look like post-puberty, where you have your epithelial cells or your luminal cells that will line these ducts, and then you have myoepithelial cells in pink, and then your adipocytes and fibroblasts here. During pregnancy, again, you have this explosion in structure dictated by hormones, and then this leads to the expansion of lactocytes, which are the cell types that are specifically responsible for producing milk. And then those components, milk components, get transported or produced and secreted into milk. You'll also notice that I'm showing an infiltration of immune cells here. I'm actually not going to talk a lot about immune cells, but I do want to remind folks listening to that there are immune cells that are also important to this process. Okay. And so these lactating mammary epithelial cells, like I mentioned, they're called lactocytes, and they are very busy cells. They're doing a lot of work. And so here's another sort of cartoon that is showing this, where this is showing a lactocyte here. And you have sort of this basal membrane here, where you have myoepithelial cells supporting these lactocytes. And then you have the alveolar lumen, where the milk components will actually be secreted. And I really like this image because it really highlights the complexity of what is actually going on in these cells, how many things that they're making, right? They're synthesizing and making a tremendous amount of protein, transporting and even de novo synthesizing some lipids, milk synthesis of lactose, which is really important here, as well as maintaining osmotic balance. I really like showing these images. This is a scanning electron micrograph. I think that really highlights some of the really nice structures and sort of crowded nature of these cells really beautifully, where you have a tremendous amount of sort of the Golgi and the rough endoplasmic reticulum, which you can see here. And then also down here, this is another example of an image where you can see these lipid droplets that are being produced and then secreted at that apical membrane of these cells. And so there's a lot going on in these cells. And so as someone that studies these cells, I try to always keep this in mind because while we do know a lot about what is happening in these cells, there are still some things that are unknown in terms of there's still some things that are unknown. And so I also think these cells have a lot of unique properties that support the production of milk components. And so, for example, there's tight control of milk production, like I mentioned in the previous slide. And there's also a production of a huge array of things. And so micronutrients, cytokines, human milk oligosaccharides, which I'll talk a little bit about later, as well as lactose. And then there are some interesting things about these cells. So they are binucleated. So some fraction of these cells are actually binucleated, much like what you might find in, I think, the liver, for example. There's some really beautiful work that has followed up on this and showed that endoreplication in these cells can actually lead to these heterogeneous cell types. And so I want you to think back to, for example, this image, right, where, not all of these cells are the same, right? They actually are doing a lot of different things. And you might get heterogeneous populations because of this. And so here you can see some of these really nice binucleated cells in these images. And then I think one thing that I really wanted to point out is that was really exciting about this particular study is that they showed that sort of this stress associated binucleation was actually really important for milk production and appears to be critical for milk production. So, how do we study cellular mechanisms of lactation? I am mostly interested in studying this in the context of humans, but of course, we know a lot about lactation across a lot of different species. And so here I'm showing a really nice image from a review article that shows, different structures, like how the mammary glands are different across species, so human, mouse, and bovine models. Similar cell types, but of course the organization is a little bit different, and the composition of the produced milk is also different. Um, but I think there are some features that are conserved across species, and so that's also always worth keeping in mind when creating model systems for studying lactation. But there are also some differences. So this is a really beautiful some really beautiful organoid work from Dr. Ronner's lab at Tufts. Where she has developed organoids, mammary organoids, from a bunch of different species. So here I'm showing images from cow, goat, rabbit, and rat. And what you can see is that, they respond to something like a ROCK inhibitor very differently, and then also have sort of different morphologies as well. And so while there are many conserved features of lactation across different species, it is also worth pursuing the study of lactation in humans specifically, because there can be some differences. As you can imagine, sampling the lactating breast is quite difficult. And so one way that we can potentially get around this is to actually leverage the cells that are in human breast milk. And so this is just a really nice image that kind of breaks down the fraction in total breast milk of the different components that you might get, where, for example, lactose is highly prevalent, proteins, of course, and fatty acids and fats. But I want to draw your attention to cells, right? There are actually lots of cells in breast milk, and these can be very useful for studying some of the fundamental biology of lactation. And that's what I'm hoping to convince you of in the first bullet point of this talk. And so this is some unpublished data from my lab, but there have been some lots of others that have generated data like this as well, where we can see that cells that are isolated from milk, and these are cells that are just shed into milk from the lactating mammary gland, these cells are alive, and millions of cells can be isolated. And so here I'm showing the number of live cells per mil of milk, stratified by 4 months less than 4 months postpartum, 4 to 12 months postpartum, and greater than 1 year. And what you can see is that even at the greater than 1 year time points, we can still isolate many, many millions of cells to study. These cells can be really dynamic and responsive to both maternal and infant factors. And so this is from a study that was looking at differences in cells that could be retrieved from fresh milk, transitional milk, and mature milk. And here are a few different immune cell markers here. And what you can see is that, of course, there are some differences between colostrum and transitional as well as mature milk in terms of just overall percentage of cells that are expressing these markers. And then overall, if you're looking at just the immune cell compartment, right, this also will change over time. And then this is another example of a study that actually showed that you can get differences in immune cell populations in milk in colostrum healthy and then also during infection. And so again, milk composition will be dynamic over the course of lactation and can be responsive to these factors. You can also take these cells and coax them to form organoids and on-chip structures, which can be really valuable for studying human lactation and things, for example, like drug transport and drug exposure. And so this is one of the earlier examples of this, a paper from 2011, which, I think it might have been one of the first to show that you could actually take these cells from human breast milk, grow them on top of matrigel, and actually get formation of these sort of spheroid-like structures. And then when you actually treat them with prolactin, you can actually get production of certain milk components, and this is staining for beta-casein, alpha-lactalbumin, and then I think a marker for milk fat globules. And so you can see that when prolactin is around, they were able to actually do this. And so there's also some really beautiful newer methods to do this as well. And I want to highlight one over here. So this is an on-chip 3D bioprinted model that actually has these programmed in ductal structures that can then be lined with human cells from breast milk. And then you can see, of course, that these are expressing a lot of the markers that you would expect to see, and then also have nice ductal and sort of branching structures as well. And one thing I want to highlight is that, and these are just two examples of publications and groups that have shown that you can that you can create these types of models with cells from human breast milk. There's, of course, more than what I'm showing here. But one thing I did want to highlight between these two different model systems is that in an organoid model system, this is a closed system. And so you'll see that, you will, even if you're generating more ductal and lobular-like structures, it's still a closed system. And so you will have secretion of milk or maybe production of milk components towards the center of that organoid unless you were to flip it outside. And then the big difference here is that this is an open system, which, as you can imagine, the actual lactating breast is an open system. And so this might allow for more faithful recapitulation of certain features or modeling of certain features of the lactating breast. Okay, so I hope that I was able to convince you that cells can be cultured from breast milk and that they can be used to study human lactation. And now I want to go ahead and talk about how we can use single cell methods to understand these cells and their basic biology. Okay, so to start, I want to talk about some of the work from my lab. And so this is a study where we wanted to use single cell RNA sequencing, which is an approach that lets you measure and quantify the expression of all of the transcripts that are present in a cell at a single cell level. And so this is an approach that has been really valuable for studying biological systems and really uncovering heterogeneity and how this might translate to interesting and useful features in these systems. And so the question that we wanted to ask with this study was to see how single cells in breast milk change over time and how heterogeneous they are. And so what you can see here is an example from our study where we were able to collect breast milk from 15 donors, seven of whom had greater than 3 time points, because we also wanted to see how these cells might change with time postpartum. And we were able to generate data on about just under 50,000 single cells. And here I'm showing a 2D representation of this data colored by all the different cell types that we were able to identify. So you can see, of course, a bunch of different types of immune cells where the majority of immune cells are actually macrophages. And then when you look at the total number of cells, most of the cells in breast milk are epithelial cells, which matches with prior literature. And we identified 2 different types of epithelial cells. And this agreed with another study from the Twigger lab, which came out in 2022, right around the same time that as right before our study, which also showed that there was 2 different types of epithelial cells. And then I put a few different citations of other studies here as well that also agree on these epithelial cell types, which was really exciting to see. And so we were able to really highlight some of the heterogeneity that is present in these samples. And then here I'm showing an image of the cell types here. This is a dot plot that shows expression of genes. And then here are the different genes on the x-axis, where you can see that each population of cells has unique sort of signature of expression of these genes that can define what those cell populations are. And so this allowed us to then call these cells luminal cell 1 and luminal cell 2, or lactocyte cells 1 and 2. And so this is what I'll refer to for these epithelial cells for the rest of the discussion today. Okay. There's also been some really nice comparative analyses. So, there, as you might have, as I showed you in this slide, there are quite a few single cell RNA sequencing studies on human breast milk now. And as you can imagine, there's even more in the context of breast cancer. And so this is from a recent article that came out that really compared a lot of different breast data sets to try and actually harmonize some of the cell typing and the naming around these cells. And for the purposes of this talk, what I really wanted to highlight is that this is a box that shows this. So this is a few different markers, genes for each of these different cell types from these different studies. And here are the cell types up here. So myopithelial lactogenic or hormone sensing, where hormone sensing are the cells that are more responsive to hormones. And what you can see is that the lactocyte cells do appear to be quite distinct, and even when compared to other studies. Okay, and so just to zoom in on the lactocyte population, and so this was from our study. And so what you can see is this is just another 2D representation of our single cell data where you can see our LC1 cells here, and then our LC2 cells could be further subdivided into smaller subsets of epithelial cells. And then again, you can see the genes that define these. And this is really important because if you have subsets of cells in a given tissue, in this case, a liquid biopsy that can represent that tissue, it can tell you a little bit more about the function of these cells and how they might be different, and if they are doing different functions or performing different functions in the context of lactation. And so that's the next question that we wanted to ask. And so we took the marker genes that define each of these different epithelial cells and we ran a pathway analysis on them. And what you can see here are some of the results of that analysis as a heat map. And so each one of these indicates the overall score for an indicated pathway. And then we've annotated each pathway based on what this pathway is involved in. So cell cycling, metabolism, transport, or for example, translation or immune related. And that is what is in this colorful track down here. And then each of these is our individual cell types. And so what you can see is that we do have some grouping of cells that are more important or sort of maybe have more of a role in metabolism and biosynthesis, some that are cycling, some that might be more important in the context, mostly LC1s, in the context of structure, and then some that are more important for translation and possibly inflammation-related activities. We then also look at the abundance of these cell types over time postpartum, since as I mentioned earlier, that was one of the original goals of the study. I'm not going through everything in that paper, but please check this paper out if you're interested in looking at more of our data from this study. But I really just wanted to highlight that when we look at the sample proportion of all of these different epithelial subsets in our breast milk as a function of time postpartum, either early, transitional, mature, or weeks to even years beyond into lactation, can see that the overall diversity of lactocytes increases with time, which could suggest some sort of specialization of these cells as lactation continues. But I do also want to highlight that our this sort of secretory population is kind of the highest population overall, except for in our later time points generally. Okay. So I think I walked through, I hope I've convinced you that we can use single cell RNA-seq to really understand the diversity of epithelial cells and lactocytes in human breast milk and the lactating mammary gland. And one of the other things I wanted to highlight is that we can also leverage that data type with orthogonal data that can be collected on human breast milk to learn interesting things about what these cells might be doing. And so that's what we wanted to do with the next study that I'm going to tell you about. And so so one of the things I mentioned early on in this talk is that lactocytes beyond beyond the production of sort of what you might think about as from milk component perspective, like lactose and caseins and sort of fats, These cells also make human milk oligosaccharides. These are sugars that are really important for for infant development and sort of helping to shape the infant gut. Um, and as you can imagine, the production of these HMOs is really important from an infant health perspective. And so understanding how these human milk oligosaccharides or HMOs are actually made by these lactocytes is really fundamental to their underlying biology. And so here I'm just showing an image that shows sort of some of the building blocks of these HMOs, and then also some of the known genes that we think are important for the production of these HMOs, broken out by type 1 or type 2 HMOs. Um, so the question we wanted to ask is, we have all this beautiful single cell data. Can we actually leverage paired HMO abundance data with our single cell RNA sequencing data to figure out genes or gene programs or pathways that are important for the production of HMOs and discern whether or not they're different in our two different epithelial subtypes, our LC1s or our LC2s? I kind of already covered this, but HMOs are these glycans that are really fundamental to infant health. And you can see a few examples of the monosaccharides that these oligosaccharides are built from here. Um, and so I'll move on to what the actual study design was. And this is also, I'm not going to go through every figure from this paper because this is also published now. And so please please check this out if you're interested. And I'm always available for questions if you want to ask questions. So for this study, what we did is we leveraged all of the publicly available single cell data sets that we could get our hands on the raw data for at the time. You'll notice that this is only three, but in a previous slide, I showed that there were more. We just weren't able to access the raw data from those studies at the time when we did all the work for this study. And so we're including three different studies here. We merged and harmonized all of those single cell data sets together. This was led by a postdoc, Sarah Kate Nyquist over here, and then also a graduate student in my group, Lacia. And so once we had this merged data set, which you can see here, where we very clearly were able to identify LC1s and LC2s across all of these data sets, we were then also able to work with Dr. Lars Bode to generate human milk oligosaccharide data to actually quantify this in our matched samples, which was really exciting. And so I want to give a shout out to the rest of the studies team because this would not have been possible without their support, including Alicia Jane Twigger and also Jane Martin Carley. Okay, so what did we want to do? So again, just to reframe you in the context of why I think this is important to talk about for this group. So there are two epithelial subtypes in human breast milk, and one of the things that my lab is really trying to understand is how these cells might be functionally different. And so the first question we wanted to ask in this study is, do we see differential expression of these human milk oligosaccharide genes in our LC1 versus our LC2 cells? And so here I'm showing a dot plot of expression of those genes. If you remember back to an earlier image that I showed you that we think are important, that we know are important for the production of some milk oligosaccharides. And then if they're being expressed in the different cell types in our harmonized single cell atlas across all of the breast milk samples that we were able to pull from the literature. And what you can see is that there are some genes that appear to be more up in our LC1 cells and some that appear to be more upregulated in our LC2 cells, and then some that are just not significant or not expressed to an appreciable degree. Another way to visualize this is actually as a log fold 2 change of our LC1s over our LC2s. And then this is actually colored for each gene from the study of origin. And what's really nice is that we see pretty good agreement across all three studies for the expression of genes that appear to be predominantly expressed in our LC1 compartment, and then ones that appear to be more expressed in our LC2. This doesn't mean that some of them are not are not expressed in LC1s, but definitely are a little bit higher in our LC2 population. Okay, so what we can take away from this is that overall, when we just look at gene expression, this suggests that there are some differences in HMO gene expression between our LC1s and our LC2s, albeit most of the genes are expressed for HMOs in our LC1 population. Okay, but it gets complicated, right? For folks who study human breast milk, you know that everything changes with time postpartum. Remember back to the abundance plot that I showed you earlier in this talk. And so then the next question we can ask is, well, let's look at the genes in our LC1s and our LC2s and then order them by expression in terms of week postpartum, as long as we have that data. And what you can see is, of course, there is a lot of variability in terms of the expression of these genes, both as a function of sample and as a function of time postpartum. Okay. The next question we could ask beyond just, are these genes actually expressed in our LC1s or LC2s, is we can ask if they're co-expressed with other genes that we know are really important for the production of milk components. And so that's what we did that's what I'm showing in this plot. And so so we have known genes that are important for our HMO biosynthesis. We've pulled some genes that are important for lactose synthesis, and then some genes that are important for the production of milk fat globules. Um, and then for each sample, this is a pairwise plot showing between each gene. So this would be for this gene and this gene. And then each dot is the number of samples within a comparison. And then the color is the proportion of samples with an association or a co-expression between those two genes. And so what you can see, so something like, for example, that's really this dark green would be highly correlated, but then also, since the circle is very big, it's present in a lot of the samples that we were able to have in this data set. So this plot tells you a lot about the variability in these data sets as well as the co-expression at the same time. And so what you can see is that in our LC, and this is for what we can see in our LC1 cells, and here I've sort of annotated our HMO biosynthesis or our lactose synthesis or our milk fat globules. You can see that there are some modules of genes that are sort of co-expressed across many samples in these data. And that when we actually look in our LC2 population, the co-expression structure changes slightly. And also there's sort of tighter co-expression of genes in lactose synthesis and milk fat globule synthesis in our LC2s as compared to our LC1s. And so this type of analysis can tell us a lot about sort of how these two cell types might be regulating these processes slightly differently, which again is pointing to putative different functions of these cell types. So if you remember earlier on, I told you that we collected HMO data. So here is the HMO data. And so this was, again, collected by Dr. Lars Bode's lab. And so you can see this is weeks postpartum. And I think we collected for the top 19 most abundant HMOs, which are shown in these different colors and patterns here. And then you can see the normalized HMO concentration as a function of weeks postpartum in the data set. And again, remember, we have matched single-cell RNA sequencing data for this data set. And this generally matches trends that have been previously talked about and discussed in the literature, which was great to see as well. Okay. And so I just annotate a few of those, right? So these ones stay the same, and then some that decrease and some that increase. And so that all checks out, which is really great. So now my lab does a lot of bioinformatics work in developing bioinformatics pipelines. And so whenever you have two different data sets, the question is always, how can we leverage these two data sets to really better understand a complex system? So in this case, the genes that might be important for production of HMOs and LC1s versus LC2s. And again, those are our two different lactocyte populations in breast milk. And so there's kind of two ways you can do this. You could take a discovery-driven approach where you analyze your data set separately and look for overlapping conclusions. You could use like supervised and unsupervised methods to look for patterns across your variables of interest. So this would be like clustering or machine learning approaches. This is a data set that doesn't necessarily fit super well in the machine learning kind of approaches that you might be thinking of because you need a lot more samples to really be powered to do those approaches. You can also use matched data to guide a supervised analysis, or you can fit into a prior knowledge base. So you can map your data types into an existing model system. And so this might be performing like a gene regulatory analysis or a metabolic flux analysis, something like that. And so really at the end of the day, how do we combine things to make better things? And so the approach that we took with combining our single cell data and our HMO data was to use our match data to guide a supervised analysis. And so what does this actually look like? And so the first thing we could ask is, well, we have concentrations of all of our HMOs. And so here, this is what I'm showing on the x-axis. And then we also have single cell expression for all of our paired samples. And so we could ask, well, are there any genes that are positively or negatively associated with changes in concentration of these HMOs in milk? and what do those genes look like? And so that's what this is showing. And so this is a plot that is showing for each HMO the number of positively associated genes or the number of negatively associated genes. And then the colors of each of these bars is either these genes are shared between our LC1s and our LC2s, or they're unique to LC2s or LC1s. And I think what's really striking about this plot is that you can see that actually a lot of the genes that appear to be either positively or negatively associated with a given HMO are not shared. Most of them actually appear to be quite unique, which could suggest that these cell types, again, are using different gene programs to actually drive or support the production of HMOs, even if the enzymes that are used for each steps are the same. Um, and I think one thing I did want to point out here is that this is a lacto and fuco pentoase 1 here. And this is the HMO, at least in our data set, that had the highest number of genes that were significantly either positively or negatively associated with the production or at least the concentration of these HMOs. And so the next question we could ask is, well, we have all these genes. Are they purchased or what are they doing? Are they working in concert together? Are they enriched for enzymes? Are they enriched for transporters? Because this can help us to try to understand what's happening in the system a little bit more. And so that's what we did. And so we took the genes that were associated positively or negatively with this HMO and then clustered the top associated genes. And what you can see, this is in our LC1 and this is in our LC2. The top track is time postpartum. And then this track right here is actually the concentration of this HMO. And then you can see the genes rows that have been clustered and then also annotated by the type of molecule. And so what's really interesting about this is that, of course, there's actually differences between genes that are associated with HMO concentration in our LC1s as compared to our LC2s. And when you look at these, a lot of them are actually enzymes and transporters, and so could be important for supporting the production of HMOs, possibly differentially between these two cell types. But now the hard part of validating these results begins. So, okay. So just to summarize everything I've talked about so far before I jump into more of the next part of the talk. And so if you remember, here's our nice picture of what the mammary gland might look like, what the mammary gland looks like during lactation, indicating all the different cell types. And you can see our LC1s and our LC2s, which I've also reproduced this original UMAP representation of our single cell data here as well, where you have our LC1s here and our bigger population of LC2s. And then here, I'm just starting to fill in some of the functions and putative functions that we can get from our single cell data so that we can try to better understand what the differences might be between these two cell types. And so here's a column for LC1s, here's a column for LC2s, again, lactocyte 1, lactocyte 2. Tissue maintenance, hormone sensitive. So I have a bunch of different functions over here, which we know are important for either supporting milk production or directly producing milk components. And so based on our data so far, LC1s appear to be more important for tissue maintenance, or at least are expressing pathways and genes related to tissue maintenance, like tight junction proteins and pathways. They are also hormone sensitive. LC2s and LC1s express immune-related genes possibly involved in tissue turnover or inflammation. LC2s are our population of epithelial cells, lactocytes, that appear to have more diversity. And so that's what I'm showing over here in this image. Gene regulation, I'll fill that in the next section of this talk. And then here are a few things related to production and transport of milk components. So either production of HMOs, milk components, lipids, milk fat globules, and then something that I think this audience will be really interested in is drug transport. And so I've started to fill in some of these, but of course, this is an I guess, simplified version of this image to start, and my lab is working to try to fill this in and validate these things experimentally. Okay, so the next part of the talk, I want to talk about gene regulation, right? So most of the stuff that I've talked about so far has been descriptive, but the next question could be, well, are these LC1s and LC2s actually regulating their functions differently? And then I'm going to talk a little bit about drug transport. Okay. So before I talk about the next study that we did, I wanted to talk a little bit about single cell ATAC-seq, or ATAC stands for assay for transposase accessible chromatin. And so that's what I'm showing here. And so, everything that I've showed you so far has been transcriptomics. And so that's going to give you a picture of the mRNA that's being produced in a cell. Um, and that kind of can tell you a lot about what a cell may or may not do in the future or might be doing currently. But we know that DNA is arranged in the nucleus around histones, and there's a lot of other regulatory mechanisms happening that can tell you a lot about how a cell is maybe performing the functions that it is performing in a different way than just looking at transcriptomics. And so this is a nice image that's showing some of those. So we can look at either chromatin scaffolding, we can look at chromatin remodeling. So there are enzymes that will actually remodel the way that chromatin is structured. There's histone modifications, and then there's also transcription factor binding and DNA methylation. And all of these layers of regulation are important to control what a cell is doing and ultimately, what mRNAs and proteins it's producing, right? And so what we wanted to do is then actually generate ATAC data on single cells from human breast milk so that we could maybe better understand some of the regulatory mechanisms that might be different between our epithelial cell subsets and lactocytes. And this was really led by a student, Lacia, in my group again. And this is all also unpublished. And so, yeah, so please, I know this will be shared, but I just want to acknowledge that this is unpublished currently. This is also a part of a collaboration with Dr. Brock Christensen up here at Dartmouth College as well. And Fred Coling at our genomics core has been instrumental in helping us generate this data. There was a lot of optimization that had to be done to actually generate our multi-omic data that pairs single cell ATAC and single cell RNA-seq together due to some optimization around lysis times for actually generating this data. All right, and so what did we do? So isolated cells from human breast milk, we collected the cell pellet, and then we performed multi-omic analyses. So this is paired single cell ATAC data and then single cell RNA-seq data. And then what I'm going to show you is some of the preliminary results from Lacia on analyzing these two data sets together. I also want to note that we have, again, if you remember back to, sorry for the animation, if you remember back to this slide, DNA methylation, we also have some DNA methylation data that is being analyzed by a PhD student in Brock's lab, but I'm not going to talk about that today. But we do have that information that we're working with as part of the study as well. So this is what our study looks like. Lacia has been collecting breast milk samples along with our study coordinator, Anyeka, who's been incredible. They've both been incredible leaders of this study. These are the donors that we were able to actually generate multi-omic data on. And you can see that the age distribution is pretty tight. And then the weeks postpartum is variable, but also sort of more close to this like 20 to 30 week range. And we have a decent distribution of BMI as well as parity. And these are factors that I won't talk about today, but are important for analyzing these types of data as well. All right, so what does joint analysis of single cell ATAC data and single cell RNA-seq data look like? I'm not going to walk through everything in this slide, but suffice it to say, Lacia has established a really nice pipeline for the analysis of this data. And today I'm just really going to focus on the identification of differential genes and differential peaks, or the regions of the chromatin that are actually open based on this data type. And then asking the question of whether or not they're differentially accessible between LC1s, or lactocyte 1 and our lactocyte 2 populations. And so this is what our final data set looks like. And so this is a 2D representation of our data in RNA space, in ATAC space, or combined RNA and ATAC space. And what you can see is that, similar to other studies I showed you earlier, we recover all of the cell types that we would expect to recover, which was really exciting to see, because all the previous studies that I showed you were whole cell sequencing studies, but this is a nuclei-based sequencing study. And so we know that every one of these cells has a nucleus, and it's a really high quality data set. This is showing how our ATAC clusters actually map to our RNA clusters, which was really nice to see that we have good mapping across most of our ATAC clusters. So then what's the first question we could ask? So once we assign our epithelial or our lactocyte populations, we have identified 5 of them in this data set. You'll remember in a previous data set that I showed you, there were actually 6. We don't really identify a very clear cycling population in this study. And so that is one that is kind of missing from this data set. But we do identify LC1 and LC2 cell types, which I've annotated up here with these colors. And you can see this is a heat map of the differentially accessible peaks or the differentially accessible chromatin regions that are that we were able to identify between across each of these lactocyte populations. And this heat map really shows that there's a lot of difference across these different epithelial cell types. We can then look at these differentially accessible peaks and ask questions about them. So we can see of these peaks, what is the percentage of them for each of our lactocyte populations? Where do they fall into different regions? Do they fall into promoter regions, 5' 3' UTR regions, or other regions? And I think what's really interesting here is that our epithelials 0, 1, and 4, which again, in this data set, our 2 and our 4 are mostly LC2-like, and then our 3, 0, and 1 are LC1-like. And they appear to be very similar to each other. So our 4, our 1, and our 0. But our 2 and our 3 look to be different and really mostly fall in promoter regions. And then we can do something similar and look for, you know, if we're looking around transcription start sites, are most of these differentially accessible peaks falling within, where are they actually falling in relation to these transcriptional start sites? And again, we can see similar distributions for 0, 1, and 4, but not so much for 2 and 3. And so taken together, this can suggest that our LC1s and our LC2 lactocyte populations have distinct regulatory patterns. So this is just talking about open chromatin regions, right? This doesn't necessarily tell you about what genes are being regulated by these regions. And so to do that, we have to perform a linkage analysis. This really lets us link open chromatin regions to putative genes that might be being regulated by these regions. And so this is pictured here. And so this is what Lassia did next. And what you can see is that for each of our lactocyte populations, there's a high degree of differentially accessible genes that have no linked peak. And then there's a decent fraction of these that actually have a linked differentially accessible peak, whether it's significant or not. And when we look at these, our LC1-like cell types appear to have genes that are mostly linked to protein folding, metal ion homeostasis, and cell-cell junctions, which makes sense. And then our LC2-like cells appear to mostly be related to RNA processing and metabolism. This metabolism one was interesting, and so then we wanted to go in and ask if our LC1 and our LC2 cells, now that we've shown, at least preliminarily, that they might be regulated, might have different regulatory mechanisms that are underlying some of their functions. We wanted to look at the metabolism. And so we can infer that computationally as well. And so this is a plot showing different different metabolic, this is a plot showing different metabolism processes on the y-axis, and then whether or not these are more enriched in our LC2s as compared to our LC1s. And I think what you can see is that overall there is a slight difference shifting from our blue, which are our LC2s, and then red, which are in our LC1. Sorry, I flipped these labels around by accident. And you can see that in our LC2 population that we have more amino acid metabolism, and then in our LC1s and then our LC2s, we have increased oxidative phosphorylation and citric acid cycle. And so again, this can help us triangulate on some of the different functions that these two cell types might be performing. Ongoing work in the lab is to actually leverage some of the surface markers that we've been pulling from these data sets to actually flow sort and do some characterization of these cells. And so here I'm showing an example flow plots of some of the data that Lassia is generating where we're actually pulling out different populations. We can see that we can pull out these populations mostly marked by TLR2 and folate receptor, where we're now trying to determine if these are good markers for our LC1s and our LC2s. Okay, I promise I'm getting to the drug transport stuff. Okay, so we have some differences in our gene regulation between these two lactocytes, lactocyte subtypes, but related work related to validating this is currently in process in my lab. But another aspect, of course, that's really important to the biology of these cells is putative drug transport. And so the next question we wanted to ask with this data that we have, again, this is all single cell data, do we see differences in expression of transporters, right? There's a lot of different mechanisms that could actually lead to that can lead to the transport of drugs and molecules across the blood-milk barrier. And so a good first step is to look at the expression of some key drug transporters. And so this is an image from a really nice paper that I think summarizes what we know about a lot of these transporters in the context of lactation and whether or not they're expressed. And so we can look in our data set and ask the question whether or not these transporters are differentially expressed between our two lactocyte populations, and then also if their expression changes as a function of time postpartum. And so here's some dot plots that show that. And so you can see a bunch of the transporters here and then either our LC1s or our LC2s. And I think what is really interesting is that there are some transporters that we do see a significant difference in expression, as indicated by these little dots here that show significance between our LC1s and our LC2 population. And then we also see that some of these change as a function of time postpartum as well, including ABCG2. So we've talked about how cells in breast milk can be used to study the lactating mammary gland, and then how my lab and others have been using single cell methods to try to better understand these two lactocyte populations during lactation, and specifically looking at their different gene regulation, what they express, and then also, for example, differences in drug transporter expression. And then my lab is currently using these cells to do small molecule perturbation studies, and so I'll talk about that for the last remaining minutes of the talk. And so, small molecule perturbation data has been tremendously useful to the field of drug discovery. These are data sets where you can take usually small molecules as a function of different doses, if you'd like, and then expose cell types, and these are usually cell lines, to these compounds, and then measure some sort of response, so some sort of perturbation, whether or not that's a functional readout like proliferation, or it could be something like transcriptomics or transcriptional signature. And so that's the image that I'm showing here. And you could do this again with bulk or single cell data. And the reason that these perturbation data sets are so useful is because they're really, really valuable for training machine learning approaches for certain tasks. And so this could allow for the prediction of drug mechanisms of action, to determine how drugs or small molecules can impact underlying lactational biology, which is what we're really interested in doing in my lab, and then also putatively drug repurposing and therapeutic de-risking. If you can design compounds that actually have minimal impacts on lactocytes and therefore milk production, or vice versa for trying to discover molecules that might actually help boost milk production. Okay, and so why is this important? So it's important because I think it's women do take do take drugs during lactation, whether they have like an underlying condition and they need to be able to take something to help with that. And so we need to better understand how these compounds might impact both lactation and the composition of milk, um, to help people make those decisions in a more informed way. And so these are two studies that I really wanted to highlight because we've sort of begun to link small molecules and therapeutic exposures with changes in milk composition. And so this is from a really nice study that showed that there are changes in milk macronutrients that can be associated with maternal medications, which I'm showing in these plots here on the bottom. And then this is another study from actually one of our collaborators that showed that metabolic profiles in milk are actually associated with these forever chemicals, so these PFAS compounds, where you can see each of the metabolic profiles that they identified in milk samples from this very large cohort, and you can see that these can actually change as a function of either PFOS or PFOA. And so there's also a few others that I'm not showing data from on this slide, but I think this is sort of something that's becoming really important and should be important to the field. And so how can we actually predict if small molecules might impact milk via their impacts on lactocytes? Because at the end of the day, the lactocytes are really the ones who are producing and serving as the gatekeepers for things that end up in milk. And so, is there a way that we could rationally predict how therapeutics might actually impact milk composition and lactocyte function? And so the first thing to do is to look at these large scale perturbation data sets that already exist. And so when we look at one of the biggest ones, is called the Links Signatures database. It has a database with 21,000 small molecules, 88 cell lines, and then almost over 500,000 gene expression signatures. But lactocytes are completely absent from all of these databases. And again, most of these data are generated in the context of cell lines, and so might not be particularly helpful for this context. And so one of the things that my lab is currently doing is generating perturbation data sets with small molecules using lactocytes isolated from milk. And so we take human milk, we isolate milk-derived cells, treat them with small molecules for a certain amount of time, and then measure their transcriptome as well as viability, morphology, and then hopefully in the future, we're going to also start measuring lipid content. And so this is something also, again, I want to give a shout out to my graduate student, Lasia, who is running this, and this is also all unpublished at this point as well. Okay, and so we've done this for a small number of compounds at this point, and so this is the some pilot data that I'm showing here. So this is a principal component plot that's showing all of our transcriptomic data in response to perturbation with a few different small molecules. And I think one thing you can see is that most of these samples are grouping by the actual perturbation, which was really nice to see. These also run on multiple donors, and the data has been batch corrected for donor. And this is great because it suggests that we can actually pick out strong signatures of these molecules and how they might be impacting lactocytes. And so, for example, if we look at the impact of this is going to be ROCK inhibition, we can see that this is directly affecting ion transport and lactation. In these samples, this is showing what pathways are actually being suppressed when we treat with ROCK inhibitor some of these lactocytes. This is a drug transporter inhibitor, so ABCG2. This is I don't think this is exclusive to this inhibitor. Sorry, to this particular transporter, at least in the concentration range that we were using. But I think one thing you can appreciate is that when we treat lactocytes with KO 143, we actually do see some differences in gene expression. When we inhibit calcium signaling, we see strong decreases in pathways that are related to secondary messengers, which makes a lot of sense. And then one of our strongest perturbations was inhibition of protein synthesis, which of course would not be good for lactation. And we see a tremendous amount of genes that are impacted by treatment, specifically with cyclohexamide. And so this is a small pilot data set, but it does suggest that lactocytes are sensitive to known strong chemical perturbations and that we can recover signatures of how we think these small molecules should be acting in the transcriptomics data that we're able to generate. We can then also perform a different type of analysis with our transcriptomics data called deconvolution analysis, where we ask the question, what are the cell type proportions that are being affected in our samples? And so you can see each of our samples along the x-axis here, and then what compound they were actually treated with, and then the cell type proportion in those samples. And I think one thing I will really point out is that EGTA here treatment leads to essentially complete elimination of our LC1 population, but not our LC2 population. And so this is important because we can start to figure out how some of these compounds actually differentially affect our LC1s and our LC2s, since both of them are important for milk production and function during lactation. So we need to understand the impacts on both of these cell types. Okay. We can then ask the question, are there certain maybe drug transporters that are being affected in our perturbation data sets? And so here I'm showing some of the drug transporters that are known in the literature, and then the expression as a function of each of our small molecule, some of our treatments that I'm showing here. And what you can see is that the expression values are variable across our donors, but we do actually pull out some effects on the impact of the drug transporter expression as well. Okay, So just to bring it back to what we're doing, and so ongoing, so we're looking at more compounds and we're also trying to link viability and lipid content as well so that we can assess how these compounds are impacting those features of lactocytes too. And then eventually the goal will be to use these data sets to build predictive models that can let us figure out if small molecule compounds are going to affect milk composition and lactocyte function. Okay, so what did we talk about today? So cells in breast milk can be used to study the lactating mammary gland, and these cells are highly dynamic. These can be cultured and used to model human lactation. It's yet to be assessed whether or not LC1 and LC2 populations are accurately recapitulated in current models, and so that would be really valuable to see. We can then also use single cell methods to understand these cells and their basic biology. So who is there? So we now know that there's two populations of lactocytes, and my lab and others are trying to figure out if those cells are performing different functions. Um, and again, they do have differences in expression of drug transporter genes, and so would definitely be important to consider in the context of research for drug exposures into human breast milk. And then my lab is using perturbation studies and generating perturbations with small molecules so that we can develop tools for actually de-risking therapeutic use as well as predicting whether or not small molecules might actually impact lactocyte function. And so to bring it back to this cartoon, so this is something again that we're actively working on filling in, but just to end on this, I think for this audience in particular, particularly for like PKPB models and things like this, as well as modeling transport ex vivo, there needs to be a way to account for differences in these two lactocyte populations, as well as time postpartum. And with that, I will acknowledge everyone for listening. Thank you again for the invitation. Thank you to everyone in my lab, but a special shout out to Anyeka, who is our study coordinator that collects all of our fresh milk, and Lasia, who is our graduate student that leads all the milk work that I talked about today, as well as our funding, and then a lot of our really wonderful collaborators, a few of whom I gave a shout out to throughout this talk, but none of this would have been possible, at least especially the unpublished work that I showed you, without support from our collaborators. And so thank you so much.
Thank you so much to Dr. Goods for joining us today, and thanks to you, our audience, for watching. If you have any other topics that you'd like to hear us talk about, go ahead and let us know in the comments below, and we'll see you next time. Bye.